Bob McKown. I'm a professor in the ISAT department at JMU. And this is a research lab where we clone, express, and purify recombinant proteins for potential therapeutic use. It starts here in our freezer where we go and we get our genetically engineered strain. The bacteria has been genetically engineered so that it has a human protein in it. We take this freezer stock that we have here of this genetically engineered protein and we start growing it up. The idea is we want to have the bacteria make a human protein. From here we can take the cultured bacteria here and we bring it out to our lab. And we actually plate it out here on an agar plate. And within that bacteria has a human gene called Lacritin. We can grow the bacteria up and then purify the protein out of the bacteria. The purified protein then can be analyzed for its various properties. One of them is causing new tear secretion. Another one is antimicrobial. We send this to our collaborators at the University of Virginia, Gordon Laurie Lab, as well as our collaborators at Eastern Virginia Medical School, where we're conducting preclinical animal studies to look at a potential new therapeutic for dry eye syndrome. We inoculate continually growing cultures here. Here's a fresh media that hasn't been inoculated yet, and we let this grow overnight. We use this to inoculate bigger yet volumes of media to bulk the bacteria up. Here we actually have a culture growing of our recombinant bacteria. And from that little blast that we started with in the incubator or in the freezer, we end up growing it up to larger and larger volumes. We then harvest the cells through our centrifugation here, where we can remove the media, pellet the cells that we need to purify the protein from. This is a device here that we use to disrupt the cells. Bacteria have pretty hard cells, cell walls, and this is sound waves that we can adjust, and we actually put the bacteria underneath the probe here and give it a controlled blast of sonic waves, and that disrupts the cells and let the protein come out. The final step in our purification process is to freeze dry the protein that we have, and this is called a lyophilizer or a freeze dryer. Same principle, freeze-dried coffee. We freeze the samples in small vials, and then we, under vacuum and low temperature, this is minus 48 degrees centigrade right now, under vacuum here, it dries or removes the water overnight. And we have our purified protein in small aliquots that can then be sent off for analysis.